Hello, class. How are you? It is the 16th of the September, and it is 1:37. I'm a little bit late, but that's okay. These are the things we talked about last time on chapter 4, and I'm hoping you finish. And I'm hoping to finish chapter 4 today. So endomembrane system, I think that's where we stopped, right? Oh, come on. Come on. I said endomembrane system. I clicked on it, and this one came about. Okay, come on. Yes, endomembrane system. So we talked about that one last time, the things. So there are a few questions about endomembrane system, things getting out and inside of the cell, and endoplasmic reticulum, the function of is a ribosome, that both large subunit and small subunit. Endomembrane system. Yasmin, do you have any question, or should I go ahead lecturing? If you have any question, please ask that first. No, you're chatting. Okay. No, you're good. Okay. You're just here for the support. That's great. Awesome. Let me put myself to so. Okay. So divided into compartments where different cellular functions occur and so on. Make sure you know all of these writings as well. It's just not the pictures. I do not read the writings, and of course, word by word, a lot of times, but I don't talk about things, and I would like you to know them, know those things. Attachment of ribosomes, rough endoplasmic reticulum is a place where ribosomes are attached to that layer of a membrane inside of the cell. Okay, synthesis of protein, definitely you should know the function now, and send it to lysosomes or plasma membrane. Smooth endoplasmic reticulum, on the other hand, relatively few bounded ribosomes. They do not have much ribosome, variety of functions, synthesis, store calcium, detoxifications, and your textbook, this PowerPoint does not say lipid synthesis. So make sure you write that down in your notes. Lipid, fat synthesis, it makes fat. Okay, that happens in smooth endoplasmic reticulum. Ratio of PER to SER, rough endoplasmic reticulum to small format, depends on the cell's functions. And here they are. They're showing you a rough ER. There is a membrane inside. It's hollow. We call it lumen. Okay, they didn't show it to you. Nice picture of the transmission electron picture, and smooth ER. And these are all mitochondria. Okay. Golgi apparatus, a packaged cell material, has a cis and phase trans. So we talked about this. This is a cis toward the nucleus, and trans is toward the cell membrane. Looking at this diagram, I would not know which end is a cis and which end is a trans, but most likely this is a cis because of all of these vesicles are a little bit vesicles here. But you know, when scientists look at it, they call it cis and trans. It's because if it is facing the nucleus, the end that is facing the nucleus is cis end phase, and the trans phase is toward the cell membrane. Protein transport through a membrane system, and the membrane system, as I said it in your quiz. Sorry. I said it. It is the stuff that goes inside of the cell, right here. Well, this is outside coming extracellular fluid coming out of the cell and going inside of the cell. It is possible that can go through the endomembrane system. Okay, so when but the pathway, the structures they use is different. For example, the materials that is going inside of the cell, they can attach to the lysosome, and lysosome dismantle it, destroy it. Lysosome is a bag full of enzymes, and then those products end up in Golgi apparatus, and Golgi apparatus can package them and send them as the cell is needed. That is going inside of the cell, getting a material getting outside of the cell. Here is a protein molecule being synthesized and goes into the vesicles, and the vesicles go into the Golgi apparatus. Golgi apparatus package them, make them, and send them outside of the cell. So there are different scenarios. I hope I'm making some sense. Lysosome. Okay, membrane-bounded digestive vesicles, digestive enzymes, you should say. There are digestive enzymes inside of lysosome. Oh, I'm running out of power. Hang on. I'm running out of power. Hey. So that is the function of lysosome. Lysosome has hydrolytic enzymes, that they use water, hydrolyze molecules, hydrolysis. So you're familiar with that term, I hope. And then there are inside of lysosome plus other structures, plus other enzymes. Okay, arise from Golgi apparatus. And then contain enzymes that catalyze breakdown of macromolecules, fuse with target to initiate breakdown, and recycle old organelles as well. That is something we call it autophagy. Digest cells for material and engulf phagocytosis. Autophagy, it means, or autophagy, it means when the mitochondria, for example, it's old, what happens, a vesicle comes to eat it, and then their enzymes from lysosome digest it, break it down. I hope there is a picture. Okay, so here is a lysosome structure. Right here, a packaged material, and it goes to material that phagocytosis. You remember we talked about this. It goes through, attached to the lysosome, break it down. This is autophagy, a mitochondria, old mitochondria are being dissolved away, old damaged organelles such as mitochondria, and then lysosome attached to them, break them apart and break down the organelle, and those structures can leave the cell. Again, back to autophagy. Okay, great. Thank you. Thank you. Thank you. Yes. Okay, so microbodies, variety of enzymes breaking down membrane enclosed vesicles, peroxisomes. It has hydrogen peroxide, again, quiz question, contains enzymes involving oxidation of fatty acids. Hydrogen peroxide produced by byproduct rendered harmless by catalysis. So these are the structures. Here is a peroxisome inside of the cell, as you can see. Anyhow, vacuoles, not animal cells do not have vacuoles. Okay, so membrane-bounded structure typically found in plants and some protista. By now, you should know what are plants. You should know what are fungi. You should know what are protista. You should know, of course, animal cells. And of course, you should know bacteria. Right? If you don't know them, please ask. Whatever I say, whatever comes out of my mouth that you're not familiar with, please ask. Various functions depending on the cell type. So different types of vacuoles, central vacuoles in plant cells, of course, storage vacuole in plant cells, and contractile vacuoles in some fungi and protista. The contractile vacuole, they are the vacuoles that hold up into a lot of water. So water get inside of the cell, osmosis. You remember that? Outside of the cell is hypotonic. Inside of the cell is hypertonic. So water get inside of the cell. There is a mechanism called contractile vacuole, get that excess water and transport it and put it outside of the cell. If not, the cells will die. Here is a nucleus, central vacuoles, tonoplast. Do not worry about that term. Chloroplast, yes, worry about that term. It causes photosynthesis. Okay, and of course you have cell wall, plant cells, bacteria, fungi, they have cell wall, and some protista. Animal cells do not have cell. Mitochondria, I talked about it here and there a little bit, but we are going to finish the chapter today. Found in all types of eukaryotic cells, bound by membranes, 2 layers of membrane, outer membrane, inner membrane space between the outer membrane and inner membrane. That is called the inner membrane. It is important. Inner membrane space. And then inner membrane shaped into folds. If it is fold, the fold is called cristae. Matrix, like cytoplasm, you have it in the cytoplasm, you have it in cell, but matrix is in mitochondria. On the surface of the inner membrane and also embedded within it are proteins that carry out oxidative fast metabolism, which we will talk about it, have their own DNA and also have their own DNA. Here we go. So you have the outer membrane, the space between the outer, they call it intermembrane space. And then look at the folds. The inner membrane folds, when it folds, that is called cristae. They have their own ribosomes, they have their own DNA, they can make their own protein molecule. Look like a footprint, doesn't it? The transmission electron micrograph of it. So the folds inside, these are called cristae. And of course, right here is called the inner membrane, and the outside is called outer membrane. Okay, I think. And the matrix is the medium, the fluid. All of the fluid area in here is called matrix, right here. This is the best part. There is no cristae here. So this, all of this area right here, it is the matrix. Do it make sense? Matrix is the, if you would, is the medium of the mitochondria. Chloroplast. Okay, chloroplast organelles presents in cells of plants and some other eukaryotes like protista. Some protista have chloroplast. Fungi do not have chloroplast. Okay, surrounded by 2 membrane, contain chlorophyll for photosynthesis. Thylakoid are membranous sacs within the inner membrane, and grana are stacks of thylakoids. They have their own DNA and ribosomes. So they were bacteria someday, but they were, by the process of endosymbiosis, they were engulfed by as eukaryotic cells, ate them, and now they benefit the cells. So here are some structures of the chloroplast. Make sure you know them. And then here are the chloroplast in plant cells. Endosymbiosis theory, that's what I was talking about earlier, proposes that some present-day eukaryotic organelles evolved by symbiosis. Symbiosis means living together. 2 different species living together is called symbiosis. For example, if you have tapeworm, if I have tapeworm in my gut, if I have roundworm in my gut, the roundworm, tapeworm is different than Amir assadi-rad, different than a human being, Homo sapiens. But those worms in my gut, they depend on my food. That's called symbiosis, living together. But a pregnant mother, since the baby is same species, it's not called symbiosis. Okay, between two free living cells. One cell, a prokaryotic, was engulfed by become part of another cell, which was a precursor of modern eukaryotes. Mitochondria and chloroplast have similarities to prokaryotic cells. Yes, that's why we think endosymbiosis theory, the cells, the bacteria were engulfed by a cell, and now the bacteria inside of that cell benefit the original cell, either by photosynthesis or by metabolism, which you're going to learn when we get to metabolism. Here it is, eukaryotic cell. Here's the original cell. You know, they went inside, the chloroplast went inside, and now we have a modern eukaryotic cells, and with both of them. So this is the endosymbiosis. This diagram right here is explaining endosymbiosis theory. Okay, the other one, the cytoskeletons are not organelles. The definition of organelle is a structure that has a metabolic function which is surrounded by membrane, or it has membrane. Rough endoplasmic reticulum, it has membrane, phospholipid membrane. Smooth endoplasmic reticulum, Golgi apparatus, mitochondria, they're all organelles. Chloroplast, they're all organelles. Lysosome, they all have membrane. Peroxisome, they all have membrane around them. However, cytoskeletons do not have membrane around them. They are just pillars holding up the cell so the cell does not collapse. Just like the walls of your home. Inside the wall, there are 2x4s, 4x4s. That's what cytoskeletons are. Support the shape of the cell, keeps organelles in fixed location inside of the cell. Dynamic system, constantly forming and disassembling. Yes. 3 types of cytoskeleton. Microfilaments, which is like actin. An example of that would be actin filament. They give it to you. 2 protein chains loosely twisted together. He's talking about actin. And then movements like contraction, crawling, pinching. In case of amoeba. Then you have microtubules, largest of all. Of the 3 types, microtubules are the largest. Largest of the cytoskeleton elements, dimers of alpha and beta tubulin subunits, and then facilitates movement of cells and materials within the cells. They act like a radar track. They're like a radar track. So the vesicles, this is a vesicle transporting materials. It moves on these microtubules. And intermediate filaments, it is between the size of actin filaments and microtubules, very stable, usually not broken down at all. Okay. Here they are. Oh, sorry. Here they are. They're showing you all 3 different sizes. And this is a cell membrane. This is a cell membrane. This is a mitochondria, rough endoplasmic reticulum. And they're showing you where they are. Here is the actin filaments, the smallest of all. Here is the intermediate filaments. And here are the microtubules. They're supporting the cell. Centrosomes, good. Your textbook is putting it as, again, there is no membrane around centrosomes. There's just a bunch of protein molecules, regions surrounding centrioles in almost all animal cells, microtubules organizing center can nucleate the assembly of microtubules. Animal cells and most protists have centrioles occur in pairs. Plants and fungi usually lack centrioles. Here they are, centrioles, and you will learn that they're triplets. Remember that, microtubules, triplets. And then during cell division, when one cell becomes two cells, they're involved. We'll talk about it, centrioles. Cell movement, the movement of actin filaments, microtubules, or both of them. Cells involve, some cells crawl using actin filaments. Eukaryotic cells have cilia, 9+2, arrangement of microtubules, 9 microtubules pairs around and 2 in the center. So cilia are shorter and more numerous. Here is 9+2. Here is a cell membrane. This is a transmission electron micrograph of a cilia or flagella. At the base, it is 3+0. The base is 3, they call it basal body. Basal body is 3+0. So, but here it's 9+2 on top. And the way they move, it is almost same as your muscle contraction. When you get to zoology, we will talk about muscle contraction, but not here. Okay, so there are 9 microtubules in the parameter and 2 in the center. 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. Cilia and flagella, of course, flagella are longer. There are less number of them. Some species could have a lot of them. And then the cilia, there are many of them, and they're shorter. Eukaryotic cell wall presence in plants, fungi, and some protists. We talked about that. I already talked about that. Eukaryotic cells wall are distinct from prokaryotic cell wall chemically and structurally. Plant and protist cell wall made up of cellulose. Fungi cell wall is made up of chitin. And plant cell wall primarily and possibly cell wall. So plants can have 2 or 3 layers of cell wall. Again, right here is a wall. They're talking about different structures. I'm not too crazy about it. Just remember that plants have cell wall. Extracellular matrix, the protein molecules that are outside of a cell, is called extracellular matrix. Animal cells lack cell wall, but they have cellular matrix, which is protein molecule. Secretes and enable matrix of glycoproteins into, what does it say, space? Into space around them, yes. Collagen may be abundant. Collagen is the most abundant protein molecules on planet Earth. Put it this way. Form a protective layer over the cell surface, and integrin links the extracellular matrix to cell cytoskeleton. And hopefully a picture will come up, so we'll discuss it. Influence cell behavior as well. Here we go. So here is the actin filament inside of the cell. Here is the integrin. It connects the outside of the cell protein molecules to inside of the cell protein molecules, called integrin. Integrin has other functions. When we talk about cell membrane later on, a phospholipid bilayer and all of that, you will see it. But for now, just based on your textbook, it connects for now outside to the inside of the cell. Okay? Let's go. Very good. Your textbook has good tables. Please study them. Make sure you know this. If you have any question, let me know. It's summarizing what's going on. The only thing that is missing in here is function. Make sure all of these structures on the left-hand side, all of these structures in here, all of them. You know the functions too. Okay? I hope I'm making some sense. Cell-to-cell interactions. Yes, the cells of our body are connected to each other and they will interact with each other. So this is very important, something you need for your future classes as well. Everything I say so far, everything in this class, you do need it for your future classes as well, if you're a biology major. Okay, so cells, proteins give cells identity. Cell makes contact, read each other, yes, and react. Glycolipid, it means the glucose molecules that are attached to lipid, phospholipid molecules. Most tissues, specific cell surface markers, MHS, proteins, recognition for self and non-self cell by immune system. MHS, I have to dig in deep in how come your textbook not say it. It's anyhow, you know, different cells of your body have different MHS. The protein surface, the protein molecules on the surface of your liver are different than the protein molecules on the surface of your brain cells, than your white blood cells. So that's why when you get a transplant, somebody remove their kidney or their heart or liver and they give them to you, what happens, those MHS proteins are the culprit that reject the white blood cells of your body, goes in there and reject the kidney, reject the liver, reject it. So they try to find people who are very close to each other. The MHC is very close to each other. Yeah, anyhow, it will come to me someday. Someday. So cell-to-cell communications, adhesive junctions, mechanically attached cytoskeletons of the neighboring cells or cells to the extracellular matrix. ECM includes adherence of junctions, desmosomes and hemidesmosomes. Septate or tight junction. There is no comma between tight and junction, but anyhow, it connects the plasma membrane of adjacent cells in a sheet, no leakage. And then finally, communication junction, which is gap junction in your cardiac cells or plasmodesmata in their plant cells. Okay, chemicals or electrical signals passes directly from one cell to adjacent one, which is, you see it in your heart muscle cells, it's called gap. In plant cells, it's called plasmodesmata. So food and nutrient from root of a 300 feet tall tree goes all the way to the top. Here they are 10Q tight junctions, almost like a nut between the two cells. So materials, things do not go from one cell to the next cell. Anchors the cells to each other a little bit. And then adhesive junctions. So you have, it's almost like tight junctions and desmosomes, but it's not little bands like a rope. They're little discs, if you would. So it connects the two cells together. Okay. And then you do have the gap junctions right here. The two cells are connected together by these little channels. These two are, nothing goes from one cell to the next cell. But the last one, things must get into from one cell to the next cell. Examples of these two are your epithelial cells in your body. You don't know what epithelial cells are, but you know, the cells in your gut, the intestine. Okay, so I'll have these two. And then, of course, this is a cell, example of cell in your heart muscle cells. Plasmodesmata, same thing as this last one. But that is in plants. Plasmodesmata. Plant cells have plasmodesmata, specialized opening in their cell walls. Cytoplasm of adjoining cells are connected and function similar to gap junctions in animal cells. Okay, so that is the end of chapter 3 material. So let's go to chapter 5. Is that right? In this exam is chapter, I'd like to talk about cell membrane. Lecture the structure of membrane. Yes, phospholipid, great, good. I thought we are going to talk about cell membrane first. Are you seeing my screen? No. Hang on. Come on, share. How come you don't want to share? Okay. Good. You can see it now. So chapter 5, and these are the things we talk about, the structure of membranes, of course. The phospholipid, the membrane's foundation, proteins, multifunctional components, passive transports across membrane, active transports across membrane. We don't get to talk about it today, but we'll talk about bulk transports and endocytosis and exocytosis later on. So having said that, let's talk about the structure of membrane. Phospholipid arranged in a bilayer. The first exam, last exam, you learned the structure of phospholipid. Very important for the rest of your life, as long as you're in biology. So includes glycerol, phospholipids, and sphingolipids. We'll talk about that. Globular proteins inserted in the lipid bilayer. Yes, it's giving you a general picture of it. Fluid mosaic model. It is those protein molecules within the phospholipids, they do move. They're not stationary. Mosaic protein floats in the fluid lipid bilayer like a boat on a pond. Okay. Here's a phospholipid bilayer, cell membrane, and they have cholesterol. And then they have the protein, integrin protein. You remember that? Integral protein, another integral protein acts as a gate, opens up and closes, and molecules go in and out. And of course, the integrin will attach the outer structures to the inner structures. Okay, glycoprotein, they are extracellular matrix, the protein molecules that carbohydrates attach to them. Extracellular matrix, protein, and then you have glycoprotein again, right here, the difference of the blood type A, B, O, AB is because of these sugar molecules attached to protein molecules. And then peripheral proteins, the proteins that are inside of the cell, attached to embedded protein molecules. Okay, and then what is a fluid mosaic model says that these protein molecules are not stationary. They do move. This one, next minute is here, and that one, next minute is there. So they're not moving in a synchronized manner. They are moving randomly. And sometimes the phospholipid jumps. This one, this phospholipid jumps and goes inside of the cell membrane. Inside, there are two layers. One layer of phospholipid here, one layer of phospholipid here. So they can jump, transfer, transform Okay, cellular membrane have 4 components. Good. Your textbook put it in this respect. Let's see what they say. The phospholipid layer, flexible matrix, barrier to permeability is a boundary of the cell. Transmembrane protein, integral membrane protein, protein molecules within the phospholipid layer. Interior protein network, peripheral protein inside their protein attached, very important, attached to the phospholipid bilayer. And the last one, the surface, the cell surface marker, which is the glycoproteins and glycolipids on the surface of the cell. Okay, it's like a railroad track, one layer and one layer, and in between are the it's a phospholipid bilayer. But anyhow, let's see. Studying of the cell membrane, the transmission electron microscope and scanning electron microscope, SEM, are used to study membrane. One TEM method is embedded specimen by epoxy, less than 0.1 micrometer thick. Shaving off embedded specimens can be imaged right here. This is cell 1. This is cell 2, 2 layers of cell membrane. And in between, it is called interstitial fluid. No, I guess it's not mentioned in here, so do not worry about it. Freeze fracture microscopy. So what they do, they freeze a cell and they cut it, fracture it, then you can see pieces of the cell membrane. Please read this anyhow. Membrane lipids, lipodemic, defines the number of biological functions of lipid. 1,000 distinct lipid in cells are divided into 3 classes. Glycerol, phospholipid, sphingolipids, and sterols, which are the steroids. Okay, here's a sphingolipid. You have 2 layers of 2, come on, go up, please. These 2 are your nonpolar molecule, the tail of phospholipid. And then you have, you don't want to move, I'll move you here then. Okay, so then in the head, you have the polar hydrophilic. They love water, this structures. One of them, it does not mention. Okay, so this is the choline group. This is the glycerol group. And this is the phosphate group. Phospholipid. But they don't mention it. When you say phospholipid, they don't mention the choline either. So phospholipids spontaneously form a bilayer because of the amphipathic. Amphi, it means double. Amphibians. So the molecules have a head. The head is hydrophilic, love water. The tail does not like water, hydrophobic. That's what they call it, amphipathic. Polar head is polar and hydrophilic, and phosphate groups attaches to the head. The 2 fatty acids are roughly parallel to each other and non-polar and hydrophobic, water-fearing. Here it is, I guess, the choline, the phospholipid. Oh, okay, that was sphingolipid. So I'm sorry about that. This is a sphingolipid, structure a little bit different. Most textbooks do not talk about sphingolipid. This is a phospholipid. This is the choline group, phosphate, and glycerol. Sorry, I thought the other diagram was a phospholipid, but it was a sphingolipid. And then what happens, the 2 fatty acid, one of them, most cases, it is unsaturated fatty acid, or it could be both of them unsaturated fatty acid. In this diagram, they show that both of them are saturated fatty acid. There's a kink here. You guys see that kink? Right here, that kink, it means the cell has a unsaturated fatty acid. Okay, let's talk about this a little bit later. I think I like to go do a few things. So this is the one I did not talk about, Yasmin. If you would, Yasamin, if you would tell me on this slide, I stopped. This is where I stopped for Wednesday. Any question? Anybody has any question? Okay, I see you recording. Yes, it is happening. I hope it is recording. Okay.